A very good morning, students. This is your teacher Neelam Elawat, and today we'll start with the next topic, that is mutation and genetic code. See, from the last two videos, we have continuing the topic, that is genetic code, and I hope you have understood. The topic. And if there is any doubt, you can talk to me, right, everyone? So today we'll start what is the relation between the mutation and genetic code. Earlier also we have spoken about the mutation. So that is in continuation, or you can say in a way it will be a revision of that topic itself, right? So let's start. Mutation. As the word said, mutation means it is a change in the nucleotide sequence of DNA. Dear students, we know DNA is made up of nucleotides, isn't it? DNA is made up of 4 nucleotide bases, that is your A, T, G and C, isn't it? So any change in any of these nucleotides will result in mutation. Clear? And it is generally estimated that out of every 10 to the power 6 cell divisions, at least one mutation occurs. So mutation is nothing new. It is a very simple process that occurs, right? It may occur in the somatic cells. Somatic cells means those mutations which will not be passed from one generation to another. Means suppose if the father has, this individual A has, it will not be passed to individual B. Okay, these are the mutations which will not be passed from parents to their children. So those are referred to as somatic mutations. Then mutations may also occur in the gametes. Gametes means it may occur while the egg is being formed. It may occur while the sperms are being formed. See, egg is a female gamete and it will be produced inside the female body, while sperm is a male gamete and it will be produced inside the male, right? So mutation can also occur in egg, mutation can also occur in the sperm, and then in that case it will be passed from one generation to another. So mutations can occur in both type of cells, somatic as well as gametes. Now mutations can be classified into two broad categories. One is a point mutation, another is a Frameshift mutation. Point mutation. Let's see what is a point mutation. Now point mutation, as the name suggests, point means only one base pair will be changed by the another, right? Only one base pair will be replaced or changed by another one. It is of two types basically. First is transition, another is transversion. Transition means where purine will be changed by a purine. Transversion means where purine is changed by pyrimidine. Suppose you are taking example in case of transition, suppose the pairing was between, see, pairing always occurs between A and T, right, and G and C. This is called a normal pairing, right everyone? A is always paired with T and with the help of a double bond, double hydrogen bond, while G always pairs with C with triple hydrogen bond. Now here we have T. Suppose this T gets replaced by the C. This T gets replaced by the C. Okay. Or this C get replaced by the A. Then it is called a transition. Purine will be replaced by purine and pyrimidine will be replaced by pyrimidine. Transversion, reverse. A will be replaced by G, or T will be replaced by G and A will be replaced by C. This is a cross connection will be there. So purine will be replaced by pyrimidine. Let's see this one again. Transition and transversion. Suppose you have CG, the sequence is CGAG and GCTC, okay? So we have a pairing between A and T. So this gets replaced with GC. This is called transition. Again we have the pairing AT. but AT get reversed. T gets here and A comes here. So this is called transversion, right? This is called transversion. Second is frameshift mutation. Frameshift mutation means where the mutations may occur in one or more base pairs that are inserted or they are deleted from the DNA. अब इसमें क्या होता है students? Frameshift mutation. We know that DNA can read DNA and DNA can also read, or DNA can also be read by the mRNA. Okay, the bases which are present on DNA are read by the mRNA, right? And they are in the form of triplets, right? So if there is one extra nucleotide added into the DNA or one deleted from the DNA, that will result in the frameshift mutation. Let's see this. Suppose we have a sequence CGAG, GCAG. I have highlighted this sequence AT. Okay. Insertion means extra will be placed. Now suppose here one extra, that is TA, has been placed. Okay, here we have 4 bases, now we have 5 bases. So one extra base pair has been added. So this is called insertion. This is called insertion. In case of deletion, this base pair will be deleted. So we do not have AT here, right? We do not have AT here. So this is called frameshift mutation, and frameshift mutation can be categorized into insertion and the deletion process. Okay, so your frameshift mutation is of 2 types, insertion and deletion. Now just imagine what will be the consequences of frameshift mutation if by chance there is a mutation in the DNA. So what will be the consequence? We know DNA will be used to produce mRNA, isn't it? And then mRNA will be used to make amino acids or proteins. Okay, clear? If there is change in mRNA, this means there will be change in a amino acid, and it might be possible if the mutation is very drastic, then this amino acid can not be produced. Okay, so insertion or deletion of a base in a gene result in altered reading frame. Okay, altered reading frame हो जाएगा mRNA का reading frame change हो जाएगा और इसकी वजह से एक different protein बनेगा और possible है वो garbled protein हो, which is not required at that point of the time. The amino acid will be changed and it will build up a protein which is not at all required, right? Now suppose we are taking a sequence. Normal mRNA has a sequence AUG. This is a normal mRNA sequence, right? AUG, UCU, UGC, AAA. And the protein that will be made is methionine, serine, cysteine and lysine, right? Now just imagine that the U has been deleted. See here, U has been deleted. यहाँ से U delete हो गया. So first sequence is same. Second is not this one. Let's have this. So यहाँ से ये U delete हो गया है, right? ये U has been deleted. Now this become a pair now. CUU, okay? And then GCA and then AA. So consequently the reading frame has changed. Now we'll have a protein methionine, leucine and alanine. So these two are the proteins which were not being made earlier. But because of the change in the sequence, now these two proteins are being made. Okay, so our reading frame has changed because this U has been deleted, and thus the reading frame shift to the next base. Okay everyone, like you can read this also, UUC, UCG, this is a normal sequence. Okay, now what happens here? See, this change has occurred. Now this U has been deleted and this sequence now becomes single base, CU, okay, and this one shift further. So UCU, then CGG, then ACC. So in this manner the frame shifts from the normal to the abnormal one. Okay, and this will result in the production of an abnormal mRNA, which will produce a different kind of amino acids in comparison to what was actually required to be produced. Okay, मतलब अगर हमारे mRNA में कोई भी फ्रेम शिफ्ट म्यूटेशन हो गया है तो उसकी सीक्वेंस चेंज हो जाएगी. उसकी सीक्वेंस चेंज होने से अब हमारे पास डिफरेंट अमाइनो एसिड्स बनेंगे इन कंपैरिजन टू व्हाट वी वर एक्चुअली रिक्वायरिंग. तो यहाँ पर देखिए ओरिजिनली हमारे पास 4 प्रोटीन्स अमाइनो एसिड्स बन रहे थे, मिथियोनिन, सेरीन, सिस्टीन एंड लाइसिन. बट यहाँ पर हमारे पास 3 बन रहे हैं. ओनली वन इज़ द ओल्ड वन एंड दीज 2 आर द न्यू ones and they are not required at this point of the time. Now this mutation can be caused by various factors like physical factors, physical agents. UV light can cause the mutations, X-rays can cause mutations, visible light can also cause mutation, and heat. Apart from this, there are certain chemicals which can also lead to the mutations like 5-bromouracil, 2-aminopurine, nitrous acid and acridine dyes. So dear students, when you are handling such chemicals, you should be very, very careful so that your skin does not come in contact with them, right? You are not in direct contact with these chemicals because they are mutagens. They can cause the mutations in the DNA, right? So I hope you've understood everything. Right. So let's come to the homework. First of all, you will make notes of this topic, right? And you will read the page number related to this topic. Okay, you will read the page number from your NCERT and you will underline all the one-word answers. You will try to make short questions and you will highlight all the important words from this page, right? Okay everyone, bye. That's all from my side.